Hello, my name is Raphael Kim, and I'm here to present our work, Surfacing Livingness in Microbial Displays, a design taxonomy for human-computer interaction. The term livingness is broadly defined as the condition or the quality of having life and being alive. Physical displays of livingness include movement, growth, reproduction, color generation, and many more. In our paper, we explore how designers can make these types of livingness from non-human organisms more noticeable to the human senses, a process which we call surfacing. And for interaction designers working with living media, surfacing livingness is an important strategy. Previous works in the field have shown that surfacing livingness can offer many advantages. It can communicate the functional status of living artifacts, whilst encouraging a more empathetic and mutually beneficial interactions to form between the human and the non-human agents. That said, however, when it comes to life forms that are microscopic, surfacing their livingness can be challenging, which reduces our abilities to notice the organisms in sufficient and empathetic ways. Many of them often appear too small and too slow, while some are difficult to cultivate, are perceived negatively, and can be pathogenic. In response, we have developed a taxonomy of surfacing mechanisms that designers could use to overcome these microbial constraints and to strategize their ways of designing. To achieve this, we first began with systematic collection and screening of living artifacts, sourced mainly from three major databases: ACM Digital Library, Springer, and IEEE. From this process, a total of 45 living artifacts were identified. The intention here was to obtain a snapshot and an overview of different microbial explorations that had been undertaken in HCI. Over a 6-month period, we began the analysis of the collected artifacts to first identify and to name the surfacing mechanisms that were present in their designs. Overall, we found 6 types or levels of surfacing mechanisms. Level 1 is canvassing. We define it as the process of framing a habitat for microbes. Common examples of canvases include petri dishes and bioreactors, which effectively provide a platform for microbes to live and to express their livingness over time. In HCI, from transparent materials such as plastic and glass, to opaque and traditional ones such as wood, all with different sizes and shapes, various materials and forms of canvassing have been explored. They suggested how changing material properties of the canvas could help towards altering and crafting of the quality and meanings of microbial displays. Similarly, as our case study on Flavobacteria showed, adjusting the openness of the canvas for the microbes to be exposed and situated closely within their natural habitat could also offer alternative ways of displaying microbes to surface hidden relationships between the microbes and the ecological assemblages. Level 2 is marking. We define it as the act of putting colored lines, shapes, or patterns on the surface of microbial displays. Markings help to surface livingness by providing a visual cue for human observers to notice and to understand the living aesthetics of organisms. A single elastic band placed around a jar of sourdough is an example of marking found in our daily lives, a strategy that helps us to notice the slow and the gradual changes resulting from microbial metabolism. In HCI, from digitally overlaid temporal markings that indicate changes of the past, present, and the future, to background and accent markings that help to highlight and contextualize microbial displays, markings have served as a practical and mostly non-invasive way of surfacing livingness. Level 3 is magnifying. Through magnifying, often through the help of a microscope and other amplification devices, humans are able to notice microbes and their behaviors that would normally be imperceptible to the naked human senses. Careful adjustments of magnification levels can also help designers to tune the quality of microbial displays, as it can refine the microbe to appear with certain speeds and resolution. In our case study on Flavobacteria, for example, demonstrated how the introduction of a new surfacing mechanism in the form of magnification can shift the quality of our experiences of microbial livingness. Flavobacteria are fast swimmers, being able to move twice their body length per second. By exploring magnification to surface these movements, designers can potentially alter the dynamics of human microbe interaction from ambient to responsive. Level 4 is translating. Translating is the act of rendering livingness of microbes into another language that humans would be able to better perceive and to understand. In fermentation, for example, the tuning of the needle of a pressure gauge or the changing colors of pH sensors are examples of translation that helps us to better notice the metabolic activities of microbes. In HCI, digital renderings have been commonly used to translate metabolic activities and growth patterns of microbes. They have also been used to encourage empathy from observers of microbial displays through humanizing of microbial behaviors and carefully crafted artistic renderings. Level 5 is nudging, which involves physical manipulation of microbes to surface their livingness. Nudging often involves exposing microbes to various types of environmental, chemical and mechanical stimuli, and through nudging we invite and sometimes impose microbes to shift their physiology and behavior. Incubating microbes with elevated temperature to speed up microbial growth is an example of nudging observed in microbiology. In HCI, various forms of nudging have been implemented, including mechanical agitations, light-induced human microbe interaction, a food-driven pattern formation, and many more. These nudging mechanisms can be adjusted in terms of their intensities to effectively tune the quality of microbial displays. And lastly, level 6 is molecular programming, which involves addition, removal, enhancement, and alterations of DNA. Through molecular programming, various phenotypes can be generated from the microbes that could help surface their livingness. Although there are only a limited number of examples in HCI that use molecular programming, it can potentially offer highly specific means of surfacing livingness. However, it may also bring ethical challenges given the invasive nature of the technique that could compromise the integrity of the microorganisms. To put all the levels together, here is the complete taxonomy. And to summarize, our paper makes two major contributions in HCI. Firstly, the taxonomy offers a diverse range of surfacing mechanisms which can be used individually or in combination. As demonstrated by the living artifacts analyzed, this feature enables designers to address the diverse range of surfacing constraints posed by microbes, making them more visible, encouraging us to be in harmony with their more than human temporalities, as well as soliciting a more empathetic behaviors from us. Secondly, the taxonomy can be used in flexible ways to meet the values of diverse stakeholders of living artifacts. Designers can adjust each level of the taxonomy and to switch from one level to the other, enabling them to create new meanings, perceptions, experiences, and non-anthropocentric alternatives. To conclude, we have presented a framework to surface livingness in microbial displays, a taxonomy for HCI that designers can implement towards fostering a more sustainable relationship between humans and the microbial world.